want to start by thanking the organizers for the opportunity to talk here. So I'm going to be talking about a collaboration between my lab and Yang Zhang's to do functional annotations this year for CAFA, and also the beyond part is what we can do to really bring these things into a useful form for experimentalists. And so I want to focus attention on one particular set of problems that you run into when we're trying to do functional annotations, and that is that we almost all rely in one way or another on sequence homology between targets of annotation and characterized targets to do these annotations. But we know, depending on the metric, the number may vary, but dropping below 60% identity, maybe you can go a little lower, things start to get really dicey trying to transfer annotations based on sequence homology. And to show how depressing this really ought to be, we took a random sampling of 10,000 protein sequences from Unipark and just made a histogram of their sequence identity to the nearest experimentally annotated homolog. And this looks really bad because definitely most of the mass is below this 0.6 identity where you start to see problems, and the peak of this distribution is right around 30% sequence identity. So the way that we wanted to deal with this was to design a pipeline that would be as robust as possible to these low sequence identity targets. These are probably the equivalent of free modeling in CASP for CAFA. And so the pipeline we have is a three-part pipeline. It contains one component based on sequence transfer, one based on protein-protein interactions, and one based on structure. And so the workflow that you're going to go into, I unfortunately can't really reach. Does the pointer show up if I'm? Oh, great, I can see it. Okay, yes, it does show up. Okay, so the pipeline that one would follow if you have a query sequence is, first of all, that query sequence goes directly into a sequence-based annotation module, I'll show that in a minute, and one based on protein-protein interactions, which again uses the sequence directly. And then, if you're not lucky enough to have a protein structure, and we're usually not, this goes into a protein structure prediction program, either ITaser for homology modeling or Quark for free modeling. And then I'll show that in more detail on the next slide. This gets used in sort of an analogy to sequence-based annotations where we do a structure-based search for homologs and annotate the protein based on that. And the output of all of this is a consensus Go term prediction from all three of these pipelines. And also the structure pipeline produces as a byproduct annotations on EC terms and ligand binding sites and functional regions of proteins. So I want to talk first about the structural side of it because it's the one that's most unusual, I think, in this pipeline. And so what we do to annotate these proteins, given a query structure, is to first do a global similarity search, alignments against all of the proteins in the PDB that have experimental annotations, and then identify both global structural homologs and also local homologs that share similar motifs to proteins in the BioLip database that have annotated functional sites. And then we're going to pull annotations based on both this global and local similarity to give you the EC predictions, the active site predictions, and the focus for CAFA, the Go term predictions. The actual math that falls into this, and I don't want to go into too much of the blood and gore on this. You can read the paper if you're interested in it. But we develop a consensus score based on a distance metric from all of the structural alignment hits. This takes into account a weighting of the local structure component, a global structure component, and one based on sequence identity. So from this, you can get confidence scores for Go term annotations. I'm always in this talk going to be denoting a given Go term as lambda. You'll get confidence scores for any of the Go term annotations that you might apply. Now, as I'll show in a couple slides, this method performs pretty well on its own, but it can't do everything by itself. And in particular, you're going to be completely out of luck if you cannot find any good structural hits for a particular protein. So we've complemented this with a sequence-based model and also a protein-protein interaction model. And so the function we use to calculate our confidence scores for the sequence-based model actually looks stupidly simple, but it turns out to work pretty well. And so what we're effectively doing is just taking a sequence identity weighted fraction of the set of blast hits and psi-blast hits that we get for a given protein that have a given Go term. And that defines the confidence score for that Go term. And we add on top of this something a little more unusual. This is the protein-protein interaction component. Again, this is pretty simple right now, at least in the version that we used in CAFA, and we've already made some improvements on it. But the idea here was that for some categories of annotation, and this turns out, spoiler alert, to work especially well for the cellular component Go terms, there is a guilt by association that you can get from interaction partners. And so for a query sequence, we are going to find the nearest blast hit that shows up in the string database and then assign Go terms based on how frequently they show up in the interaction partners of that protein. So these are the methods. One quick note I want to make, I forgot to say 2 slides ago, just going back for 1 minute to the structure because I think it's a really important point. When we started doing this, I thought we would really be limited by the accuracy of the structure predictions, but that turns out not to be correct because whatever challenges protein structure prediction programs currently have, they are good enough to get us good results in these alignments, in that we don't get any better results by actually using the crystal structure when we know it than we do by using the ITacer models or the quark models. Okay, so I've been yakking about how our methods work. Let's see how they do. Oh, one more important detail. How do we pull these things together into a consensus? And the answer, again, is pretty stupidly simple, but has the advantage of having very few free parameters. We are just taking a geometrically weighted average over the confidence scores we get from the 3 pipelines. And in the particular implementation we used in CAFA, the weighting on the structure term increased as the sequence identity to the nearest homolog went down. It was always 1 minus that identity, and the weights on the other 2 terms were always 1. So not a whole lot of complexity there. So let's see how this works now. I want to define a particular way that we're going to do these tests. We've done these benchmarks in a lot of systems, but I'll focus on one that my lab is really interested in, and that is annotation of the E. coli K12 proteome. So we took about 1,200 proteins that actually have reliable annotations in this proteome, and then we try annotating them, and we give the maximum F-measure scores, as is done in CAFA, but with one important wrinkle. As we go along the x-axis from right to left, we are progressively restricting the sequence identity of the nearest homolog that one is allowed to use in this annotation. So as we go from right to left, we're simulating those cases where you don't have any nearby targets to use in sequence-based annotation. And so these are the 3 baseline predictors in CAFA and how they behave in this setup. You can see that the BLAST and PSI-BLAST predictors end up with some information content at high sequence identity cutoffs, but as you restrict them to not having good sequence identity targets available, they drop to be below noise by the time you're getting to 30%. This naive method is just predicting everything just on the basis of how much each Go term shows up in the annotations. And so just to compare these before we get to our method with a good sequence-based method, we applied GoFDR, which had performed very well in the last CAFA competition, to this same kind of challenge, and it performs a lot better than these other sequence-based methods. But again, we get this progressive decay of accuracy, or of overall performance, I should say. This isn't strictly accuracy, as we go to lower sequence identity cutoffs. Now, something really interesting happens when I just bring up the structure component of our pipeline, COFACTOR. And so just the structure component of this pipeline starts off with performance that's not any better than the sequence-based methods, worse than some of them, but it is highly robust to these decreasing sequence identity cutoffs. And in fact, this line does have a slope. It's just very small. And so you can see that by the time you're getting down to the 20 or 30% range, this is outperforming all of the sequence-based methods because you're seeing that structural information is conserved with function over much longer distances than sequence information is. And now we can capture that in our annotations. And so if we add in the other 2 components of this hybrid model that we have, you can see our sequence-based model actually performs pretty well at high identity, but like all of the other ones, it starts to drop off as you get to lower sequence identities. And the protein-protein interaction module, interestingly enough, shows that same level of robustness that the structure-based model did in terms of performing very well even at low sequence identity thresholds. But the most interesting of all is when you pull this hybrid prediction together. And so that's the complete COFACTOR prediction. It ends up outperforming all of the individual components. And this is a little surprising intuitively at first, but it is exactly what you should expect if these models are making uncorrelated errors. There's some very nice work from, say, Scott Page on this topic. And so we get 2 major takeaways from this. One is that structural information in particular is usable and carries information over longer distances than sequence does in terms of evolution. And 2, that we're making different enough mistakes with these different models that we're able to meaningfully combine them to get a better consensus prediction. And I was showing biological process Go terms for the last couple slides. The story is essentially the same for all 3 Go categories. I think the most interesting thing here is just the fact that the cellular component category has historically been a real bear to do better than naive methods on. The protein-protein interactions seem to save us on this one because it does a particularly good job there. All right, so This is partly about our predictions in CAFA, and so it's worth talking about what we did for CAFA. And so we have 3 sets of predictions in the competition. Of course, we're very anxious to see how they did. One is just our sequence and protein-protein interaction-based pipeline. This was for all the targets we could do it for because we could find structural homologs, and this was all but 7,000 of them in the competition. We applied the full, including structure, COFACTOR pipeline to 6,000 of the targets that we considered hardest in having the lowest sequence identity to the nearest annotated homolog. And then we have combined predictions where we put together the ones between 1 and 2. And I will point out the reason that we had to do the full COFACTOR pipeline on only a small set is it's really computationally expensive. The compute time for each of these, the sequence and protein-protein interaction pipelines together take less than 1 CPU hour per target. The structure predictions are slow, though. This varies wildly with protein size, but you're going to be above 100 CPU hours for a lot of targets. The structure pipeline alone, just doing the comparisons, about 20 CPU hours. So this accuracy and insight does not come without a price, but it does give a lot of additional useful information. And these times are still low enough that we have been using this to annotate whole microbial proteomes. So I'd like to spend the rest of my time saying to please think of the experimentalists. And when I say that, it sort of sounds like please think of the children. But half my group is experimental. And Go terms are very useful. They have a lot of very useful properties. But as was pointed out earlier today, you cannot typically just take a Go term and figure out exactly what a protein is going to do. So one of the wonderful things about this structure-based pipeline is it gives a lot more richness of information than just the Go terms when you're annotating a protein. And so, for example, this is the first panel of the output from the COFACTOR web server when you run it on a target. And I've also got the address of the server here if anyone's interested in trying this out on your targets. There are several other panels, but this one gives you a set of structural homologs along with predicted ligand binding sites and even identities of predicted ligands that you're then able to use to construct follow-up experiments. And then aside from this information, we also have panels that show things like the Go term hierarchy that you have for each of the aspects of Go terms that are involved here. And so just to give an idea of how useful this can be, I want to consider the case of an E. coli protein called YAIP. We know it's important from knockout studies on resistance to several types of antibiotics, but we don't really know why. And the only annotation you can get if you're looking at Go terms is that it's a predicted glucosal transferase. But if instead you look at the COFACTOR annotations, we get a richer story. We get, first of all, we're working from now a predicted structure, and we can see there's a UDP binding site over here. We can see that there's a glucose binding site over here, a couple more glucose binding sites over here, and what looks like it might be an exit channel. And so from these predictions, you can read off the story of this protein. It looks like it's a glucosal transferase that's going to polymerize UDP glucose into some kind of linear glucose polymer. And sure enough, we went ahead, we purified this protein, and it does polymerize UDP glucose. It doesn't work on other sugars. And if you mutate this residue, arginine 250, which is predicted to interact with a phosphate in the UDP glucose, you completely abolish activity. So we can take these structural predictions and we can read off the entire story of what one of these proteins is doing just based on those predictions. So just to summarize what I've shown at this point, we're seeing that hybrid models, especially by incorporating structural information, can give a large benefit in terms of function prediction. And I think very importantly, we've reached the point with protein structure prediction that it's good enough to guide this kind of annotation. So we don't just have to rely on crystal structures for that. That doesn't mean, for example, that we're predicting all of the contacts right, but we don't necessarily have to. And a side benefit of this is giving additional richness of information that's very useful also for experimental follow-up studies. And so definitely there are some near-term improvements that are possible, and we'll probably have lots more near-term improvement ideas after we see the CAFA results. In terms of, we're doing some work to further curate all of the databases that go into this, especially the binding site data. We'd like to do a better job of integrating the protein-protein interactions in particular. And we'd like to do a better job of identifying key residues. And we've also toyed with these ideas of domain decompositions that a couple folks have been talking about to really focus the model on the parts that are likely to matter. Okay, so with that, I'd like to thank the folks who did a lot of this work. Chengxin Zhang is a shared graduate student between myself and Yang Zhang, who did essentially all the computational work that I've shown today. Mehdi Ruckenpour is a postdoc in my lab that's been doing both the YAAP experimental follow-up and also a couple other fun examples that I don't have time to talk about right now unless anyone asks about them. And then, of course, Yang Zhang and I have been really partnering on this, and also members of both of our laboratories. And if anyone's interested in seeing the paper, you can find it on our website. It was in NAR a couple months ago. And I'd like to thank you all for your attention. I can take any questions you have.
Great talk, thank you. I'm very impressed by the use of structural information. I think this is a welcome addition to the CAFA and this SIG in particular. I'm wondering about its usefulness, and I hope to see those results soon because, well, no, I'm not criticizing anything.
I have extra slides if you really want to see the other cases.
So here's my question. So we have established in previous years that sequence-based methods do well both on high similarity and low similarity sequences, almost equally well through the combination of multiple hits and use of machine learning. I just wonder whether some sort of a machine learning perspective of structure can push that higher for both high and low sequence identity.
Yeah, well, so, and I think that is incidentally what you were seeing with, for example, the GoFDR case, where it did far better as you went down to the lower homology targets than the more naive methods. So I think the answer is yes, and that's definitely an area that we're starting to work on now is using machine learning methods to do a better job of pulling together all the structure information that we get. Part of the reason that the methods we used were so stupidly simple, for example, our sequence predictors, was to avoid overfitting, because we were really deathly afraid of that, and we're also afraid of that for the structure information. So I think it's something that could be highly beneficial as long as it's integrated very carefully. So yeah, wait for CAFA3, and we'll put that in. I don't know who's next.
Chair should say. Yeah.
Chair.
Hi. So yeah, I was also, so regarding the use, the addition of structural information, so two questions. One, how often does it actually crop up as useful, you know, given that, you know, TASR is a well-known structure predictor and everything, but, you know, in terms of whatever we have, there are plenty of sequences for which we really cannot predict structure, or are there? So how much information, or let's put it this way, the percentage of sequences out of all CAFA targets that were assisted by introducing structural information. Do you have an idea?
Well, I have no idea because we haven't seen the results yet, and because we did not benchmark against the old CAFA targets in particular. We were taking other protein databases to look at that. So the best I can do is say look at the improvement performance of the hybrid model over the sequence alone, and that gives you some sense of how much the structure part matters.
Okay. And the second question is, in one of your graphs, you've shown that you're going down to 20% identity. Yeah, 0.2.
Yep.
That's almost midnight zone. That's like, so that's pretty impressive, actually, if you get structure to help you in this level, or anything to help you on this level, actually.
Right. Now, one important caveat to that one point, definitely the protein-protein interactions are still robust down there. This graph was made with structures that were always modeled using a 30% sequence identity cutoff. So that means you're making your life harder than it should be over here. You're making your life possibly slightly easier over here. And that was just so we didn't remodel the entire proteome for every different cutoff. So when I'm saying we are using this sequence identity cutoff, that is for the members of all of the databases that are being used for annotation, for the BioLip hits, for all of the Go hits, for example. But the maximum allowed homology for iTaser was still 30%. So it's possibly not slightly that hard, but also there's not a whole lot of proteins that are that far down to 20%. So,
thank you.
Yep.
Hi, can you please show the slide where you calculate a score based on blast and PSI blast hits?
Yep. Yes.
So you take all blast hits and all PSI blast hits?
All significant blast hits, all significant psi blast hits.
Get the two scores and sum them by some weight.
Right, they're summed by, the weight that's being used here is the sequence identity to the query. So if we just look at this term, for example, that means you're summing in both cases over all blast hits. In the numerator, you are taking only the blast hits that have the Go term you're interested in with those sequence weights. In the other case, you're in the denominator, you're using all of the blast hits.
My question is, aren't blast hits a subset of psi blast hits, or at least in the most cases?
They don't have to be, and especially when you are getting down into lower sequence identities, you can have psi blasts be more sensitive. Oh, sorry, aren't blast hits a subset of psi blast hits? Yes, that is correct. Sorry, I had it inverted in my head. But you'll note that there is a weight term here, such that you weight the blast hits more heavily when you have high identity partner or high identity sequences to annotate with, and you weight the psi blast more heavily when you have lower identities. So it turns out that the blast is more informative when you're high sequence identities.
Thank you.
Great. Time for one more question.
Could you just please say a word about your string-based Go term predictions?
Oh, sure. I think this was the this was the one that probably took the least amount of time. So I said we're trying to do a lot more than this right now in terms of, for example, introducing a weighting of many different string entries in the same way we did for blast. But right now, really, it is just as it looks here. You take your query sequence, you blast it, you take the blast hit with the best e-value, you take all of its protein-protein interaction partners from the string database, and then you assign a confidence score for each Go term based on the fraction of those partners that had that Go term. And that's really it. And so like I said, there's a lot more I think we can do with this. But it already turns out to be a very useful hedge for some of the other methods when we put them together in the hybrid model.
Great. Let's thank our speaker one more time.